Okay. Um, Thank you. Before I get started, Praful, thank you very much for inviting me. It's actually an honor. And also, I think it's like not a huge group. So if I don't know, I don't know how you normally run it, but I would be happy if anybody wants to interrupt with a question, for example, so it's more like a real talk. So if something is unclear, um, please interrupt me. And so I will be talking about this actually recent paper um, called Thermodynamics of Darwinian Selection and Molecular Replicators. And this paper came from a more general interest of mine, which is trying to understand how the fundamental laws of physics constrain universal principles of living matter. And by the fundamental laws of physics, I'm especially thinking of the laws of thermodynamics, so which are very universal, which are do not depend on the material, what kind of matter systems are made of, most of them anyway, and don't depend on particular models. And in also in recent times, thermodynamics has moved away from studying purely macroscopic near equilibrium systems to genuinely stochastic and far from equilibrium systems with the development of so-called stochastic thermodynamics, which some of you are familiar with. And so stochastic thermodynamics now is able to consider the thermodynamic laws that govern systems that are far from equilibrium, that are possibly microscopic, molecular scale, and undergo stochastic fluctuations, and also that exchange matter, energy, and information with their environments. And for that reason, I actually think that stochastic thermodynamics is a promising tool for trying to answer some questions about the origin of life. I also think it has some limitations, which I can talk about maybe later. So one of the ways that stochastic thermodynamics has been applied, especially to modern biology, is via the concept of functional trade-offs. So generally, a functional trade-off is some kind of inequality that governs the relationship between some measure of functional performance, maybe speed, accuracy are typical ones, and thermodynamic cost. Typically, how much free energy is needed to achieve that level of functional performance. And if this lets us do things like calculate efficiencies, constrain what is possible and impossible in the universe, and also to design more optimal systems. And so in prior work in the field, this has been applied to a lot of, I would call maybe modern biological functions. So things like copying information stored in polymers very accurately, running various kinds of molecular machines like molecular pumps and walkers and things like that, and doing things like sensing and signal transduction and so on. But one of the things that I'm interested in, and actually I'm kind of now in the last year of Marie Curie Fellowship, which is trying to study this question, is whether we could kind of use similar trade-offs to say something about even proto-biological functions. And so even more kind of basic functions you might need to sustain a living system. And one of the functions I'm interested in is the ability to undergo Darwinian evolution, right? Or yeah, to basically to evolve. This led to a paper that actually just came out, although it's been in some kind of preprint form for many years and went through many rounds of submissions and so on. Has a long history, but it just came out in a special issue in Philosophical Transactions B that actually is very related to questions concerning the origin of life. So let me talk about the question considered in this work, and it basically concerns the thermodynamics of selection or Darwinian selection. And what do I mean by that? By that I mean that Darwinian selection happens when we have a population of replicators, and the replicators that have higher fitness outcompete the ones with lower fitness. Now, you might think this is almost like a truism or tautology, but as I'll mention, it's not a truism or tautology. It does not always have to happen. There's limits on how strong actually selection can be. And we can place that limit by using something called the critical selection coefficient. So selection coefficient in general is a measure of the relative difference between the fitness of two replicators, zero indicating that there's no difference. And the critical selection coefficient is basically the smallest difference that in a given system we can differentiate between. And as I mentioned, there are limits to the strength of selection. It's not always that the fitter replicator will win. And a couple of very well-known limits are one is provided by neutral theory, which basically says that the selection coefficient that can be selected is got to be greater than 1 over the effective population size. And this has to do with the noise inherent in sampling in very small populations. In very small populations, uh, Praful might have a higher fitness than me, but just due to noise, my offspring might win. And another well-known bound on the strength of selection was derived by Eigen. This is called the error catastrophe, which basically says that you can't have mutation rates be too high. So fitness might be high, but if the mutation rate is higher than the selection coefficient, then you cannot maintain the fitter replicator in the population at higher concentrations. And in this result, I'll give you kind of a preview of what we derive, is this bound that says that the selection coefficient has to be greater than e to the minus, and then I have the sigma star. And so S, as I mentioned, is the selection coefficient, the relative fitness difference between two replicators. And sigma star is the Gibbs free energy dissipated when the fitter replicator makes a copy of itself in steady state. Okay, so it's measured in units of kT per copy, per single copying event. So, I already kind of gave you some motivation, but we might ask, why do we care about a result like this? Well, first, we might want to, first, it makes a prediction, that could be tested, and maybe it's an interesting constraint on modern replicators. But I also think it's interesting from the point of view of abiogenesis and the origin of life. And there's this beautiful paper that I think many of you know, which kind of summarized various important thresholds that had to be overcome or transitions that had to be overcome during the process of the origin of life. And one of them is this Darwinian threshold, maybe one of the most important ones, where presumably some kind of prebiotic chemistry transition to chemistry that can undergo Darwinian evolution. And so one of the things I'm interested in is, can we place some kind of bounds on, let's say, the free energy flux that had to be coming into the system, or the driving that had to be coming into either Earth or some other system somewhere else, maybe in the universe, in order to allow for selection to happen. Okay, so let me now get into the main results. I'll begin by talking about the setup. So we're going to consider some kind of chemical reaction volume, and it's going to have n different types of replicators. And the reaction volume is also going to dilute out at some dilution rate phi. Now, I will mention that in the paper, I also study the case actually of a closed reactor, or a reactor maybe without a fixed dilution rate, but I'll just focus on this simple case for the purposes of this talk. And I also want to mention that, I often think of this flow reactor. I mean, this looks like a kind of a very laboratory-like setup, but we can also imagine something like a mountain lake, right? That's has streams flowing in and streams flowing out and remains at a constant volume. So we can actually very easily, I think it's a very natural kind of setting to imagine non-equilibrium chemical systems that are basically fed with a stream with nutrients and are drained by another downstream. And we're going to consider deterministic concentrations of the different replicators in this reactor in steady state for the purposes of this talk. And by deterministic, I mean that we're going to ignore fluctuations. So each of the replicators undergoes this first order autocatalytic reaction. And here the AIs and the alpha I's are basically the um, other reactants, the substrates, and the betas indicate the stoichiometry of waste products. And to this reaction, we can assign the Gibbs free energy. Sometimes it's also called affinity. And basically, this is how much free energy is dissipated per a single reaction event. Okay, so and this is a very kind of fundamental measure of, let's say, thermodynamic driving or thermodynamic dissipation. For example, it means that we could couple this replication event to an uphill, a thermodynamically uphill reaction and drive it as long as the uphill driving was less than sigma. So this is going to be the kind of cost or resource we're going to consider. And we're going to also think of the system in steady state. And in steady state, we're going to have this curly J, which is the autocatalytic current, which is basically the net current across this reaction. Okay, so how often X's are produced due to this reaction. And it's going to have to balance the outflow, right? This is going to be the steady state condition. Okay, so this is the basic setup. I don't know if there's any questions.
Just
Yeah,
a quick
please. Huh?
clarification. Is beta always less than alpha, or you don't care about such an assumption?
No, 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 no. I mean, actually, usually, like, they're not even gonna be right. Usually, these are gonna be integers. So these are stoichiometric coefficients, and usually, like, they're not even gonna be non-zero for the same i's, right? So like, alpha I might be one for like a high energy substrate, maybe, and beta I will be one for like a waste product.
Maybe overall, somehow, the idea is that, of course, you have more, since you have more X on the right-hand side, then the overall numbers of the other species should decrease, I guess, something like this. I don't know.
Well, like we overall, like we would imagine that the mass, for example, the mass would be
Yeah, exactly.
conserved,
Yeah,
right?
yeah.
But that could mean like, for example, maybe there's many monomers. Uh, so like also like that mass could go into X. So a good example would be like alpha I could be like 10 monomers if X is a polymer of 10. Maybe I'll actually show some diagrams and we'll see what this might look like. So I'm gonna make two assumptions in my derivation, and I'm gonna actually justify these assumptions to some extent. So the first one is that we can assign um, we can assign basically a one-way flux to this reaction. So we can assign a quantity of how often this reaction happens forward and how often this reaction happens backward. And the net current, so that curly J I wrote, is of course going to be the difference of these two quantities. But we want these quantities to satisfy two properties. And let me show you these properties, but let me also tell you that I think this is important, that like I'm generally not considering only elementary reaction schemes like this, where like let's say X binds to some A and then turns A into a copy of itself. That would be like an elementary reaction in my diagram. I'm considering actually arbitrarily complicated reaction schemes. For the purposes of this paper, I'm considering ones that are linear. But for example, this would be a much more detailed version of a replication reaction where we are modeling the binding events, the conversion, and the unbinding, which would be like a kind of Michaelis-Menten scheme. But we can also have more complicated schemes. So this is, for example, the replication of a dimer, so a two polymer, where it first binds, let's say, to one antisense monomer, then it binds to another antisense monomer, then those monomers bind together, then it splits off from the dimer, right? And so this is kind of important because a lot of existing results in non-equilibrium thermodynamics, they apply most naturally to these kind of elementary reactions, but this is not a very realistic scheme for how actual replicators copy themselves. So for we're gonna say that that basically we can assign a forward and backward uh, current, and that these forward and backward currents, they're going to have a mass action like form, where they're going to have some effective forward rate constant and some effective reverse rate constant that is actually can depend on the dilution rate itself in steady state, and it can also depend on the concentration of different substrates and waste products, which I'm going to emit. And we're going to say that these forward and backward currents satisfy something like the flux force inequality. And basically, this means that the ratio of the forward and backward, the log ratio of the forward and backward fluxes, is going to be a lower bound on the affinity. Okay. Now, I'm actually going to derive it a bit later, show that this holds for elementary and non-elementary replicators, but that's basically the two formal results that we're going to use. So I thought there was a question maybe, but so the next thing I'm going to have to introduce is a notion of fitness. And to me, this was actually kind of one of the interesting things going into this project, is I realized that there's in biology, in evolutionary biology, there's many different definitions of fitness that apply in different contexts. And it's actually it's a pretty difficult concept to pin down formally. And there's even this quote by the biologist Stearns that says fitness is something everyone understands but nobody can define. Or I should rather say, like, maybe that can is defined differently in different settings. I'm going to propose actually two different definitions that turn out to be equivalent of the fitness of a molecular replicator. So the first one, which applies in steady state, is that the fitness is going to be the critical dilution rate. So it's basically the dilution rate at the maximum dilution rate beyond which the replicator cannot maintain itself in the reactor. So if the dilution rate is larger than the fitness, the replicator must be extinct. And if it's smaller, then the replicator may be present. And we can show that the fitness is then using these results, which is this is the expression of the autocatalytic flux. This is the steady state condition. We can show that the fitness is defined via this implicit equation, which we can solve, right? Where this is the effective rate constant at a given dilution rate, and this is the equation. And so if we apply this to this definition, we can then solve it numerically, for example. And here I demonstrated for a simple Michaelis-Menten scheme. You don't have to worry about the colors mean they're concentrations of the different basically intermediate species. But as we increase the dilution rates, we see that the concentrations in the reactor drop monotonically. And as we hit the dilution rate, then what's called washout happens. There's basically the concentration plummets to zero. Okay. Now, by the way, you might wonder, like, why don't I just define fitness as like per capita replication rate? That might be one way, natural way to define it. Well, the per capita replication rate would be this J divided by X, right? This is this is the growth rate. This is X is the number of the amount of replicator. But we see that in steady state, this would just be equal to the dilution rate, which makes sense. The per capita growth rate in steady state has to be equal to the rate of leaving the reactor. And so, for instance, all the replicators in the reactor would, by that definition, have the same fitness. So that doesn't really, that's not actually a very useful definition.
So I have a quick question, Artemy. So so I'm still trying to understand your assumptions. So on one hand, you say that you are not considering only elementary reactions, and you can have this more special type of, for example, networks. But then when you choose the fluxes, it's just like, something times x and the minus x squared. So I'm confused, like, because from the choice of flux, it just seems that you do have one elementary reaction for which you have mass action kinetics, right? But then you say that you don't.
Can you, I think I might answer your question with the next slide. So
Okay,
hold that question. Okay.
Yeah.
So let me just say briefly that there's another way to define fitness, which is often used in biology, which is a non-steady state one, which is called invasion fitness. And basically, the invasion fitness says if I have a closed system and I put a tiny little bit of a replicator in that closed system, what is its initial exponential growth rate? And it turns out that if we compute that, then we can actually show that it's equivalent, it's the same as the critical dilution rate is defined via this implicit reaction equation. And also we can validate that sorry, this should say actually this is time, not dilution rates on the x-axis. But as time goes on, we see that the concentration grows linearly, sorry, exponentially. This is a log plot, exponentially with the fitness. So this is, I just want to say this definition I think is actually very general, and it covers a few different notions of fitness. But we are going to mainly focus on this one. So it's fitness as the critical dilution rate, the largest dilution rate that a replicator can sustain.
Excuse me, can I ask
In terms of, yes. Mm-hmm.
Yeah, it's Sylvain Charlat speaking. I'm trying to understand whether your definition of fitness actually captures the basic notion that you initially introduced, which is this relative differences between the different replicators in the system. So we expect that fitness is something that measures how good you are compared to a reference.
Mm-hmm.
That's the usual thinking in evolutionary biology. Is this captured by your definition? I think I missed this connection.
So, I mean, I would kind of say that the initial part was, actually, you can imagine that it holds or doesn't hold for any definition of fitness. So it's sort of like if I give you a fitness, could I ask whether one survives and one doesn't survive? Is that, I mean, I guess that would be a consistent definition of fitness. Now, in this case, if we imagine increasing the dilution rate, for example, then we see that the replicators that would go extinct first are ones with lower fitness. So, that's a way of saying that you have to kind of think about an operational context. An operational context here is, yeah, if we increase the dilution rate, then it does hold.
Okay. Thank you.
So let me touch upon Praful's question, because here I'm just gonna show that if we have an elementary replicator like this, then we have the current is just given by the mass action across the forward and backward reactions. This would be the J plus, and this would be the J minus. We can show that the flux force, what I call the flux force inequality, actually holds with equality. So if we calculate the thermodynamic definition of the Gibbs free energy reaction and we plug in these numbers, we can verify that they're equal to each other. This is kind of known, this is well known from kind of basic non-equilibrium thermodynamics of chemical systems. And if we have that the notion of fitness, actually, it doesn't depend, for example, on the dilution rate. It just depends on it's basically the pseudo rate constant of that forward reaction. So it depends on the rate constant of the forward reaction times the concentration of the substrates that enter into the reaction, raised to their stoichiometric coefficient. So it's just the forward mass action, basically rate constant. Now, if we have a non-elementary reaction, such as the ones I showed here, where there's multiple steps in series that are required to be completed before the replication happens, we can write it in terms of this complicated reaction scheme, right? Where we have X and it goes through some number of intermediate products, and at the end it spits out X plus X, while also consuming some substrates along the way, and possibly releasing waste products. So these might be, for example, monomers that join in. So we can show that we can still define the forward and backward pseudo rate constants so that the requirements are satisfied. So basically that there's a mass action like current, there's the flux force inequality is satisfied, and the fitness is given by the forward rate constant. And the effective pseudo rate constants are given by actually this linear algebraic equation that's stated in terms of the matrix of the forward and backward coefficients. So I'm not going to go into it, but it's, you know, described in detail in the paper. And I think this is kind of actually one of the maybe one of the interesting contributions of the paper is that it derives actually for a complicated reaction scheme, sort of in a flow reactor, possibly with dilution, it derives the effective rate constants that satisfy these results.
Sorry, I have one quick question. For the elementary replicator, is the fitness depends on the beta I or not? Because it's counterintuitive. It doesn't depend on beta I.
Right, that's a good question. So the fitness doesn't depend on the stoichiometry of waste products. Yeah. And one way to think about that is that, you know, the fitness is what happens either at very small initial concentrations or very close to washout. So right before you're going extinct in the flow reactor. So that means your concentration is very, very low, right? The concentration of your replicator is very low. And when the concentration of your replicator is very low, the reverse reaction is basically negligible because it goes like x squared in concentration. So the reverse reaction, which is the one that depends on the stoichiometry of the waste products, because they're going backwards, it doesn't matter for the definition of fitness.
And a follow-up question is that general true for all the other more complicated scheme that all these reverse beta are irrelevant? Because if that's the case, we can assume they are zero. Is that let's see.
Oh, well, I'm not sure we can assume that there's zero. So I think that's not necessarily true for the more complex replicators.
Okay.
But I have to check.
I'll read your manuscript first and then maybe follow up. Yes, thank you.
Great. Yes, great. That's an interesting question. I don't think I'm not sure it's generally true. So let me just say, combining the assumptions that I made before and also the definition of fitness, the first main result says the following, that the Gibbs free energy of reaction in steady state per replication event is bounded by this quantity. And we can kind of think of this inequality as basically saying that like the dissipation per reapplication event is bounded by this function of the actual growth rate, where the growth rate is, let's say, the autocatalytic flux per capita, the actual growth rate divided by the maximum growth rate. And so as the actual growth rate approaches the maximal growth rate, which is fitness, or as we can also say in steady state, as the actual dilution rate approaches the maximum dilution rate, this has to diverge. So we can imagine a few different replicators with different fitnesses. And as we increase the growth rate, their dissipation diverges. Sorry, as we increase the dilution rate and hence the growth rate, the rate at which they have to grow to keep up with dilution, their dissipation diverges and eventually they go extinct. And this touches upon a question that was asked earlier about like does it connect to this Darwinian notion of selection? In some sense it does, in the sense that replicators with lower fitness go extinct before replicators with higher fitness. Great. So in the interest of time, I'm not going to go over the derivation of this result, but I'll talk about one simple implication of it, which is, I would say, is kind of the second main result. So imagine that we have one replicator with fitness F that is not extinct.
Sorry, Artemy, can I ask one quick question? So why do you so phi is the dilution rate, right? Why do you call phi the growth rate, like actual growth rate in your last slide?
Yeah, sorry, maybe that's confusing. But in steady state, the dilution rate is equal to the per capita growth rate.
Okay, thank you. Yeah, I understand.
Does that make sense? And from that point of view, the fitness, which is the maximum dilution rate, is also like the maximum growth rate.
I see. Thanks.
So given two replicators in steady state, imagine we have one replicator that's not extinct, and so it has fitness F, right? And now let's imagine another replicator that is driven to extinction. So it has basically no concentration in steady state. And so remember that means that the second replicator is driven to extinction if the dilution rate is greater than its fitness. That's how we defined the fitness basically in terms of the maximum dilution rate, right? And so if we just plug that in here, what we see is that the dissipation of the fitter replicator is bounded by this inequality, and this is just minus log of the selection coefficient between the two replicators. So basically what this is saying is that if I want to drive the system so that I can distinguish for a fitness difference of S, then I have to dissipate at least minus log S. And as S goes to 0, so as the fitness difference gets smaller and smaller and smaller, the dissipation gets larger and larger and larger, right? So to see like a vanishing fitness, to see, quote unquote, a vanishing fitness difference, I have to dissipate an infinite amount. There's also like, maybe I should have maybe started by just giving a very high level intuition why we expect a bound like this. So we expect a bound like this because we know that in equilibrium, all of the sigmas vanish, right? That's the definition of equilibrium. All the Gibbs free energy reactions vanish in equilibrium. We also know in equilibrium there's no selection. And because everything, all the replicators is just going to be present in their equilibrium concentrations. And we also know that in the purely irreversible regime, where this diverges, there's only going to be one replicator that can win, which is the best replicator. So we kind of expect that there's an intermediate regime where the more we dissipate, the more we can select kind of continuously from not being able to select at all to being just able to select a lot. So we can just rearrange this inequality to put up the inequality I showed earlier. Okay. And interestingly, sorry, I know I'm starting to go over time, so I'll just finish in a couple more minutes. This we can compare to some, for example, real life data. So we can compare it to, well, we don't know the selection strength, but we know the amount of dissipation in some real life replicators, including things like prions, ligation of an RNA molecule, like in the famous experiment by Lincoln and Joyce, and things like self-replicating peptides. And we can basically see in these systems how much selection is possible. So for example, when there's different prion strains competing with each other, how big does the fitness difference have to be before one definitely pushes out the other one? And I want to emphasize that this kind of bound, it's complementary to other bounds. Like it holds even in infinite population sizes, and it holds even without mutation and so on. So it's complementary to other bounds on selection that I discussed earlier, like the Kimura's bound or Eigen's bound. I will say, maybe we can discuss it in questions if it's interesting, that this is a very small, like these are very, very small energy scales. And so one of the difficulties is scaling up this to larger systems. In the paper, I work out a detailed example where I basically consider chemostat, and there's some substrate that comes in, and there's 4 different types of replicators. They undergo what's called competitive exclusion, where they compete for this one substrate. And I basically show that if we solve the equations, actually in this case the bounds are tight. So we can predict when the different replicators go extinct as a function of the dilution rate, either by solving, the exact dynamical equations and finding the steady state, or equivalently, we can do it just by thinking about their dissipation, and we get the same exact predictions showing when basically the different replicators push each other to extinction. Maybe I won't go through this example in detail, but it's in the paper, and I think it kind of helps to illustrate what's happening. I can also, if there's questions, I can go over it. So let me just finish by saying that in the paper, I also consider things like collective autocatalysis. So this is autocatalytic sets. So something like where one replicator helps another, makes, basically synthesizes a copy of the next one in a cycle. And there's a very simple real-world example of this, which is self-complementary polymers. Sorry, complementary polymers, not self-complementary, but sense and antisense polymers, where basically one polymer catalyzes the synthesis of another polymer, and then the other polymer catalyzes the synthesis of the first. So this is not a simple self-replicator. This is a collective self-replicator. And basically, I show that you can still define fitness in the same way, and you still have the same, basically the same bounds, but now they concern the free energy dissipated by each step in the cycle. So each step in the cycle must obey these bounds on the fitness and replication rates. Great. So if you're interested, please ask me a question and go to the paper. Let me just summarize what I think the main contributions of the paper are. First, I showed how to derive essentially effective mass action kinetics that are also thermodynamically meaningful for arbitrarily complex autocatalytic reaction schemes. I propose a general definition of fitness for molecular replicators, which is defined in terms of the simplicit equation for the forward rate constant. I use it to derive thermodynamic bounds on selection and growth, and I show how to extend to some simple cross-catalytic cycles. There's some constraints that I talk about in the paper. So the things I think could be interesting to work on. First, I think introducing stochasticity into this picture would change the story quite a bit and in interesting ways. I also didn't talk about mutations, so I didn't talk about how, for example, Darwinian, I just talked about the selection part, but Darwinian evolution requires both mutations and selection, so it's kind of interesting to think of whether there's some interesting thermodynamics to say about once we introduce mutations. There's it's interesting to extend the formal results to more complex topologies of reaction schemes. So, for example, branching reaction schemes, non-cyclic collective autocatalytic sets, and finally, I'm also interested in different kinds of replicators. So everything I said here relies on first-order replication, but it may be possible to generalize some of these results to other kinds of replicators, such as the hypercycle. And with that, I thank you, and I apologize for going over time a little bit.